Well, this is the second part of our series on linkage, lecture series. And in the first part, we kind of talked about some basic terminology, the concept of linkage, the fact that you have multiple loci being carried on the same piece of DNA, same chromosome. And I said we were going to then look at two different types of linkage. One we're going to call complete linkage, which is what this part is about. And then part three, we'll come back and talk about the more common thing, which is incomplete linkage, the one that really will set most of our problem sets. But trying to understand linkage, I think complete linkage is probably the best place to start because it gives it's kind of the extreme, right? You often start at the extremes and then talk about something else that's more complicated after you look at the simple extremes. So let's go back to what we're dealing with here. If we just take a line, a continuum between independent assortment, right? Independent assortment is the idea that when we go through meiosis, what happens on one set of chromosomes up here, right, is completely different than what happens often someplace else. They're independent. So basic idea is that you might have two loci. This one has got allele, looks like they're going to have big A and little a here. And these are homologs. They would, of course, replicate and do all those sorts of things to make sisters before you did this, but that's a form. And then we assume that locus B is off on some other chromosome, maybe off on this blue one over here or something. It's a non-homologous chromosome, right? So they're off somewhere. Now, if you go through independent assortment, of course, that's what you get, right? Now, what we're going to do now is we're going to say that's not what's happening. What's happening now is that we have both loci fairly close together on the same chromosome. Okay, so we no longer have this fully independent situation going on. What we have, we're going to call it complete linkage. We're going to say that these two loci always travel together, that these two are just stuck together like, you can think of it as a large logging chain if you want, and links, and they're so strong that you can't break the links, right? This forms. Now what's going to happen here, instead of dealing with two sets of homologs, so we don't have a set of dyads, a tetrad here and a separate tetrad. We have one set of tetrads, right, going like this. And so those travel along and go together. That's complete linkage. All right. So just some ideas about complete linkage. The basic idea is that no recombination can occur. Okay, there are no recombination between the two loci. So the definition of linkage is they're on the same chromosome, right? So in this case, they're so close that you don't have any recombination. Either they are very close, okay, which is one way. They're just so close that they just, one follows right after the other. And we'll talk about how that works sometimes later when we really look at where genes are located, but that's one of the ways. The other is that sometimes there are crossover inhibitors, things that stop the process of crossing over and recombination. Male fruit flies, for our purposes, have this. Not the females. Females can cross over during meiosis all you want, but in males is actually something that sort of inhibits the process by failing to produce the right polymerases, things to recombine it and all that. Okay, so we're going to say that male fruit flies don't have recombination. Actually, one of the lab exercises that you'll do is where you'll try it with females and you'll see that there's recombination. Try the same system with males and you won't see recombination and you'll get different outcomes. So male, for our purposes, have complete linkage. Other places it can occur around centromeres. You often don't have a lot of breakage around centromeres, and you often don't have a lot of breakage around the telomeres, the ends. And there's just particular areas of chromosomes that get protected. They have their own little guard systems, essentially molecular guards, that sort of stop sort of this linkage from going on. All right, now all linkage is going to be set up with the same basic type of cross. We're doing what we said was a 2-point mapping, which we're dealing with just two loci. Okay, and the cross that we're going to use for that is that we're always going to use a dihybrid het versus a complete homozygous individual over here, and usually a recessive, although if you've got some other forms. But for right now, for our purposes, we'll treat this as a complete dominant recessive system, and we're going to have a homozygous. That's only the way we're ever going to do this. Okay. And even if we do a trihybrid, it's going to be big C, little c, little c, little c, right? We'll use a trihybrid type of back cross. So we're going to do this. So you're not going to see 9-3-3-1s and all that. We're going to see very simple outcomes. All right. So under Mendel, let's go with Mendelian outcomes. We're expecting to see this set of homologs here acting completely independent of this set here, right? And we should see an outcome from that. And that's exactly what, this is a trivial cross for all of us, right? Again, Mendel's ideas as far as segregation are still there. We're crossing a het times a homozygote, expecting a 1-to-1, het times homozygote, 1-to-1. No fancy 3-to-1s or 2-to-1s or anything like that right now. We're just going to worry about that. And under Mendel, we'd expect one from whatever this genotype produces as a phenotype to one of those to one of those. So this might be tall reds, this might be tall blues, short reds, short blues, or whatever. Okay, we give equal numbers. So again, not worried about a 9-3-3-1. We're going to go with this simple outcome here. Okay. Now, this is the individual that we're concentrating on, and we're going to keep concentrating on this guy right here for this whole lecture series. This one's just a placekeeper for our purposes, right? It's not going to produce anything except to help us get through the process. In fact, some of the problems you may see in the text or on the web with female fruit flies, where you're allowed to have crossover, they won't even talk about mating it. They'll just say the eggs from the female had this combination. And so the reason being is that everything you need to see happens over here, right? So now we're in a situation where we're not going to allow the red and the blue to cross over, and therefore no recombination can occur, right? The red ones over here are going to, while they're forming, they'll form a synaptonemal complex like they should. They're just not going to cross over in any way. Okay, they're going to be prevented from doing that. So whatever's here, when you pull apart, it's the same exact copies as you go through meiosis and you end up in meiosis 2, where you're just doing this. Okay, so that's what the outcome would be. All right, if we have that. All right, so under linkage, under complete linkage, right? So we can't have any crossover. We're going to get something that looks like this. All right. Instead of getting 25% of them to be whatever this phenotype is, and 25% that one, 25% that, and that, we get 50% this and 50% that with none of these in the middle. Okay. All right. That may be a little confusing. Let me just hang with me for a moment and I'll explain why that we get that outcome. Okay, because it turns out it can be that one or another. All right. Now just to remind you, this guy right here is just a placekeeper. Okay. All it can produce is gamete. It doesn't matter how much crossover this thing does. It can't do any recombination because all you can ever produce are little a, little b, right? So technically, while there's recombination of the genetic material between the paternal and maternal, at a genotype level, there's no difference because it's only going to produce those, right? Well, those represent this little a's right here and these little b's right here in these genotypes, okay? They represent those guys right there. So for our purposes, we can just forget those. They're just placekeepers. What we're concentrating on is that you've got big A, big B, and little a, little b from this guy over here, but somehow you did not get big A, little b, or little a, big B, right? You got 0 of those, okay? Well, there are, given this genotype and this cross, there are 2 possible outcomes. Now, only one outcome can come at a time. You can't get both. I can now explain that in a minute, but one or the other will happen. So it's one or the other of these outcomes. So the big R there, okay? So really put a big star by this page so you understand, okay? It could have just as easily come out that we ended up with 50% of them, 1/2 being this genotype and 1/2 being this genotype, but none being these others. You didn't even see them, okay, at all. And again, we're not worried about this row, right? We're looking at what's being produced by this individual here, and either, in this case, he gave me a big A and a little b, or a little a and a big B, but somehow I didn't get a big A and a big B together. They didn't show up in the progeny, right? Okay, so this OR right here becomes critical. This OR comes from a new idea for us, right? That idea is called configuration. Configuration is this. Now that we have linkage, if we take this genotype, excuse me, where you have big A, little a, big B, little b, it can have 2 possible configurations, only one at a time, but it could be in either one, okay? Just depending on the cross. And you'll set up some crosses in lab and you can play with this to see how you can get to different configurations. But one of the configurations would look like this. If these little red lines are supposed to be the homologs, right? So there are these things, right? If we could put big A and big B. So the dominant, in our case, variations of the alleles are over here. So locus A, the dominant is together with locus B dominant, which means that on the other homolog, you must have little a, little b, right? Something like that. Okay, that could happen. Same genotype, absolute same genotype could be look like this. Okay, where you have big A, little b, and little a, big B. All right. Now that's going to give the same phenotype, right? Because if we're doing dominant recessive, this big A is going to dominate over that little a, this big B is going to dominate over that little b. So this would be tall reds, and this would be tall reds, exactly. But they have a different configuration, as it's called. Okay, this configuration over here is called coupled, and this configuration is called repulsed. Okay. And by idea, the coupled is that the dominants are coupled together. They're traveling together. The recessives are traveling together. They're coupled. In this one, the dominants repulse each other. They're far, they're not connected to one another. They're repulsed. Okay, so you can remember that, right? You're dating someone you like. You're coupled up. All right. You go out and you go on a blind date. And you really don't like the person. You're repulsed. Okay, so something like that. That's a silly way, but maybe that'll work. Okay, so coupled or repulsed. You'll see them when you're searching the web or something. Sometimes these are called cis and trans. So this is cis configuration. This is a trans configuration. Sounds way too much like organic chemistry to me, so I'm not going to use that term. I'll use coupled and repulsed. Now, what's important, though, is the gametes that you can get, right? Assuming we've got complete linkage, right? This one can only make two types of gametes, right? It can make big A, big B, because there's no way to get a little b with that big A, because you can't have crossover or recombination. That's the only gametes you can get from coupled, and this is the only gamete types you can get from repulsed. All right, so this gives you coupled and repulsed. So let's go back and look at these crosses and look at them in the two forms. Again, this side is pretty irrelevant. Okay, we're interested in what's happening over here, and this is just a placekeeper to get us that 1-to-1 to 1-to-1 assortment ratio. Okay, all right, let's try it with coupled. We'll just make our little figures up like this. Okay, that's our coupled. This one over here is basically irrelevant. Okay, doesn't really matter. Gametes we can produce over here are only big A, big B, because we can't get little b with big A or anything. Here we can only get this, okay? And then of course this one can only make one type. We put those together, you've got to draw one from this parent, that's A, and one from this parent to produce, C down here. So if we put this gamete with this one, we're going to get big A, big B, little b. If we put this gamete with this one, we're going to get little a, and little b, little b. Notice there's no way to get big A, little b, right, with little a, little b, or in converse. So when we do this, it's going to produce this, right? Okay. And again, this line right here of little a's and this line of little b's comes from over here. So it's not, it's not important, right? It's what's happening up here. So the reason you can't get any of these is because this guy can't cross over. There's nothing there. You cannot get big A with little b or little a with big B. There's no combination of these gametes that give us that, right? Now, if we jump to the repulse configuration, we start out like this. Again, we're assuming complete linkage. Okay, that's the rules that we have right now. We can only get these guys traveling together, big A, little b, little a, big B. Okay, so those are the only two kind of gametes we can get. If we put this gamete with this, we're going to get little a, big B, little b. And we're going to get big A, little b, little b. Okay, but we're not going to get big A, big B anywhere together, right? And so we just change it to this. We change it to this complete flop outcome, all right? Okay, that's all linkages. The point of this series of lectures was to introduce this idea that an extreme of complete linkage, which does occur, okay, and to talk about that you get two completely different outcomes, and to introduce the concept of a configuration. This genotype can be in one of two configurations. When we go and do a 3-point mapping, it can actually be in 4 possible configurations, which makes it a little more challenging. But for right now, we're just worried about this. Second thing to say is we're always doing this cross. Don't try to set up a big A, big B, little b times big A, big B, little b. There's no reason to set that kind of cross up. We can see linkage in its best form using this. So unlike when we're doing modes of inheritance, and we always wanted to use hets times hets, right, to see the biggest range, Here, we want to simplify the whole process, so we're going to use this, right? Okay, that's complete linkage. The next part of this, part 3, we're going to look at incomplete linkage. And then in part 4, we'll come back and work some basic problems from all this.